Welcome again to BioBits and Bytes. Today we're diving deep into the fascinating world of how cancer treatments interact with our immune system. We're focusing specifically on something called immunogenic cell death, or ICD.
Yeah, and you shared some really interesting papers. We've got one from PMC on radiotherapy-induced ICD biomarkers, another from Frontiers, looking at chemotherapy and targeted drugs causing ICD.
Right.
And there's a broader Frontiers review on ICD in immunotherapy generally, plus one from Genes and Immunity on high-grade glioma specifically.
And another Frontiers piece on oncolytic viruses, right? And ICD.
Exactly. And finally, a PubMed abstract about targeting ICD, and another PMC article looking at a specific gene signature related to ICD in those high-grade gliomas. So quite a bit to chew on.
Definitely. So our mission today for you, the learner, is basically to unpack all this. We want to understand, what ICD actually is, how different cancer therapies might trigger it, what the key signals are of these GAMPs they talk about, and crucially, how this knowledge could maybe help improve cancer treatments, especially for those really tough cancers like high-grade gliomas.
And we should probably touch on the complexities too, right? Some of the contradictory findings, just to give a balanced picture.
Absolutely. Okay, let's get into it then. What exactly is immunogenic cell death? Lay it out for us.
Okay, so what's fascinating here is that ICD isn't just any kind of cell death. It's a specific, regulated way for cancer cells to die.
Regulated, meaning?
It's programmed, not just like falling apart randomly. And the key thing is, when these cancer cells die in this immunogenic way, they release these danger signals. Think of them like little alarms.
Little red flags for the immune system?
Yeah.
Because normally cell death is pretty quiet, right? Cells die all the time.
Exactly. You've got it. Typical apoptosis, the usual programmed cell death.
Yeah.
It's often immunologically silent. The cleanup crew comes in, no big fuss. But ICD, it's noisy. These danger signals actively alert the immune system. They could trigger the activation of immune cells, particularly dendritic cells.
Oh, dendritic cells. Aren't they the ones that sort of present the evidence, get the immune response going?
Precisely. They're like the field generals. They get activated, grab bits of the dead cancer cell antigens, and show them to other immune cells.
Okay.
And that can kickstart a whole anti-tumor immune response. This includes activating cytotoxic T lymphocytes, the CTLs.
The killer T cells.
Those are the ones. They can then go hunt down and kill other similar cancer cells. So, yeah, ICD is basically pro-inflammatory. It's yelling, "Hey, immune system, look over here."
Got it. Okay, that makes sense. So these danger signals, the sources called them DAMPs, right? Danger-associated molecular patterns.
Yep, DAMPs. These are the key molecules that get released or exposed on the surface when a cell undergoes ICD. There are a few major ones mentioned in the articles. Maybe we start with calreticulin, CRT.
Calreticulin, yeah, I saw that one a lot. What's its deal?
So normally CRT hangs out inside the cell in a place called the endoplasmic reticulum. It's involved in making proteins fold right. But during ICD, it can actually move to the outside surface of the dying cell. And this seems really important. Some research suggests that if a tumor has low levels of CRT before treatment starts, it might actually make treatments that rely on triggering an immune response less effective. It's like the eat me signal isn't strong enough.
Huh. So having more CRT on the tumor cells beforehand could be beneficial.
It looks that way, yeah, at least in some cases. Like there was a study on non-small cell lung cancer patients getting radiotherapy.
Okay.
The ones whose tumors had higher CRT expressions actually had significantly better overall survival. It's pretty compelling.
Wow, yeah.
And the thinking is that low CRT means less effective signaling to those dendritic cells, which messes with antigen presentation and the whole immune activation cascade.
And didn't one paper mention a link to phagocytosis genes?
It did, yeah. In those same lung cancer patients getting radiation, the tumor CRT levels correlated with genes involved in phagocytosis. That's the process where immune cells engulf the dead or dying cells. So it all seems connected.
Okay, so CRT looked like a potential marker of a good response, maybe. What about the next one? Heat shock protein 70, HSP70. I definitely saw some conflicting signals there.
You picked up on that, right? HSP70's role is, well, it's complicated. Like CRT, it's a chaperone protein, usually inside the cell. During ICD, it can also move to the cell membrane.
And in the lab, that seems good.
In preclinical studies, yeah. HSP70 on the surface of dying cells can interact with receptors on dendritic cells and help activate those killer CD8 plus T cells. Seems promising.
Okay. There's always a but, isn't there?
There often is. Here's the twist. quite a few human studies actually link high levels of HSP70 with a worse prognosis in different cancers, things like osteosarcoma, head and neck cancers after chemoradiotherapy.
Wait, worse. How does that work?
Yeah, it's counterintuitive. Even in advanced cervical cancer getting chemoradiation, patients who had a complete response tended to have lower HSP70 levels before treatment started.
Ah.
And another one, breast cancer patients. If their serum HSP70 levels went up after radiotherapy, they were more likely to have the cancer come back or spread.
Wow. Okay. So something that helps activate immune cells in a dish might actually be linked to bad outcomes in people. Why the disconnect?
It's likely because HSP70 does a lot of other things inside cancer cells that are actually pro-survival for the tumor. It can help them avoid senescence, stop growing but not die. It can help them metastasize, spread,
Right.
and even interfere with how well therapy works. So just seeing HSP70 on the surface isn't the whole story. Its overall role seems highly dependent on the cancer type, the context. It's not just a simple good damp.
Okay, that makes more sense. Context is everything. It's never simple. Right, let's move on. HMGB1, high mobility group box 1, another big one in the sources.
HMGB1, yeah. So this is usually a protein found in the cell nucleus, involved in DNA stuff. But when it gets released from dying cells during ICD,
It becomes an immune signal.
a potent one actually. It acts as a strong immunostimulant, binding to receptors like TLR4, toll-like receptor 4, which is a major danger sensor on immune cells. And yes, radiotherapy has been shown to trigger HMGB1 release.
Okay, sounds good. But again, I recall the sources mentioning mixed results in the clinic.
You're remembering correctly. It's another complex one. For instance, in rectal cancer patients getting chemoradiotherapy, high levels of HMGB1 inside the nucleus were linked to a poorer response.
Inside the nucleus.
Yeah.
But isn't the released HMGB1 the important part for ICD?
Exactly. That's what some researchers pointed out. Maybe measuring nuclear HMGB1 isn't telling you about the ICD effect. It might just reflect a more aggressive tumor state before it gets released.
So location, location, location again.
Pretty much. Because, in contrast, a study on esophageal cancer found that more HMGB1 expression in the tumor microenvironment, so outside the cells after chemoradiotherapy, was linked with better survival.
Ah.
They even saw HMGB1 levels go up in the blood after treatment in those patients. And similarly, in rectal cancer, if HMGB1 moved from the nucleus to the cytoplasm before treatment, that correlated with better survival and less recurrence.
So release or movement seems key.
It seems that way. But then again, a study in lung cancer patients getting thoracic chemoradiation didn't find a link between circulating HMGB1 levels and better outcomes, though they noted their patient group was quite diverse, which could muddy the waters.
Right.
Oh, and interestingly, one study saw a slight rise in plasma HMGB1 during treatment in colorectal cancer patients, but only if they had a specific genetic profile, wild-type KRAS, not if they had the mutant form.
Wow. So HMGB1 is a biomarker. It seems super dependent on the cancer type, the treatment, where you measure it, maybe even the patient's genetics. It's like dialing in a very specific frequency.
That's a good analogy. It really highlights the challenge. And there was one more damp mentioned, ATP.
Ah, yeah, ATP, the cell's energy currency, right. How does that fit in?
Well, when cells die via ICD, they release ATP. And this released ATP can act as a chemoattractant, basically calling immature dendritic cells over to the site. Come check this out.
Okay, so it brings the generals to the battlefield.
Initially, yes. But another twist in the tumor microenvironment, there are enzymes that can quickly break down ATP into adenosine.
And adenosine is bad for immunity.
Generally, yes. Adenosine can be quite immunosuppressive. You can tell T cells and other immune cells to calm down, essentially dampening the anti-tumor response.
So ATP starts as a danger signal, but can get converted into a stand-down signal.
Exactly. The net effect really depends on that balance in the local environment. How quickly is it released versus how quickly is it converted? It adds another layer of complexity to understanding ICD's impact.
Man, it really is a complex cocktail these dying cells release. Okay, so we have these damps. Now, how do the different cancer treatments actually make the cells release them? How do they induce ICD?
Right. This is crucial for leveraging ICD therapeutically. The sources covered several angles here. Let's talk radiotherapy first.
Okay, radiation.
We already mentioned it can induce CRT exposure and HMGB1 release, but it does more. Radiation can also make tumor cells display more MHC molecules on their surface.
MHC, those are the things that present the antigens to T cells, right?
Exactly. More MHC means the T cells can see the cancer cells better. Radiation can also increase expression of death receptors on tumor cells, making them more sensitive to being killed by immune cells later.
So it flags them for destruction.
Yeah, and it can also boost the production of certain signaling molecules like chemokines and interferon gamma, which help attract T cells into the tumor itself. So radiation can sort of warm up the tumor environment immunologically.
That sounds quite multifaceted. What about chemo, standard chemotherapy drugs?
Yep, certain conventional chemo drugs are known ICD inducers. Not all of them, but a specific subset. Anthracyclines, like doxorubicin, we mentioned that one with CRT.
Right.
Some DNA-damaging agents, though, interestingly, cisplatin seems to be an exception there, proteasome inhibitors, and also paclitaxel.
So drugs that have been used for ages might actually have this hidden immune benefit.
Potentially, yes. Doxorubicin, for example, not only triggers CRT exposure, but can work synergistically with other immune-boosting agents. It's not just about killing cells directly.
Fascinating. And it's not just the old school stuff, right? Targeted therapies, too.
Correct. The newer generation of targeted drugs.
Mm.
Some of them also seem capable of inducing ICD. The papers mentioned examples like cetuximab, crizotinib, ceritinib, ibrutinib.
Cetuximab used in colorectal cancer.
That's the one. It seems to work in part by causing stress in the endoplasmic reticulum, that protein-folding factory inside the cell. And this ER stress can promote the cancer cells being engulfed by dendritic cells, initiating that ICD cascade.
So even highly specific drugs can have these broader immune effects.
Really?
And the viruses, oncolytic viruses.
OVs. These are really interesting. They're engineered or naturally selected to preferentially infect and replicate inside cancer cells, eventually causing them to burst open lysis.
And that bursting releases the damps.
Exactly. The cell lysis itself can be a strong trigger for ICD. And what's really exciting is combining OVs with other immunotherapies. Think OVs plus CAR T cells, for instance. The idea is the OV kills cells, releases damps, attracts immune cells, and then the CAR T cells have a better environment to work in.
A 1-2 punch.
Sort of, yeah.
Yeah.
And different OVs might induce different patterns of ICD markers, which is another area of active research.
Okay. And finally, you mentioned physical therapies beyond radiation.
Right. Photodynamic therapy, PDT, was mentioned. That uses light-activated drugs. The process generates reactive oxygen species, ROS, and causes ER stress, leading to damp release.
ROS, those can damage cells quite badly.
They can. And also hyperthermia using heat. This can also induce ICD features, improve antigen presentation by the tumor cells, and help T cells get into the tumor. So quite a range of modalities can potentially tap into this ICD pathway.
It really seems like there are many roads to potentially triggering this beneficial immune response. Now, things got even more complex when the sources started talking about other ways cells can die that might also be immunogenic, beyond just classic ICD associated with apoptosis.
Yeah. Heading down the rabbit hole of regulated cell death. It's a rapidly evolving field. We're realizing that apoptosis isn't the only programmed way cells die, and several other pathways can also release damps and provoke inflammation, kind of blurring the lines.
So ICD might be part of a bigger family of immunostimulatory cell death.
You could think of it that way. For example, there's necroptosis. It's a programmed form of necrosis, messy cell death, but it's regulated by specific proteins like RIPK1, ROPK3, MLKL. It leads to cell membrane rupture, releasing damps and inflammatory stuff. And importantly, necroptotic cells can trigger those tumor-specific killer T-cell responses.
Okay, so programmed but messy, and still talks to the immune system. What else?
Ferroptosis. This one's iron-dependent, involves lipid peroxidation damage to fats in the cell membrane, and also releases damps. Early ferroptotic cells might even help dendritic cells mature. And interestingly, radiotherapy might induce ferroptosis by generating those lipid ROS we mentioned.
Oh, iron-dependent death. That's specific.
It is. Then there's cuproptosis, even newer, copper-dependent, linked to mitochondria. HMGB1 might be involved there too. And others mentioned were parthenotos, linked to DNA damage and PR, alkalotosis, pH-dependent, pyroptosis mediated by gasdermins, very inflammatory, lysosomal cell death, and even autosis, linked to autophagy.
Wow. Okay, my head is spinning slightly. It feels like the cell has this whole toolkit of ways to die, and many of them have the potential to signal danger to the immune system.
That's a great way to put it. And understanding which pathways are triggered by which therapies in which cancers is really key. This brings us nicely to that really challenging cancer type high-grade glioma, GBM. The sources went into specifics there.
Right, GBM. You mentioned it has this very immunosuppressive tumor microenvironment, makes it hard for treatments, especially immunotherapies, to work.
Exactly. So the hope is that maybe inducing ICD could help overcome that suppression, make the tumor hot immunologically. Studies have looked at damp release and GBM cell cultures after radiation or the chemo drug temozolomide.
And what did they find?
Well, radiation showed some damp release, but interestingly, temozolomide, which is a standard chemo for GBM, seemed to induce only limited ICD markers in those lab studies.
Hmm. That's maybe not what you'd hope for from a standard drug.
It highlights the challenges.
Yeah.
But then there was that other study looking at a gene signature related to ICD in high-grade glioma patients.
Yes, the one with a counterintuitive finding.
That's the one. They found that patients whose tumors had an upregulated ICD gene signature actually had worse outcomes and seemed less likely to respond well to immune checkpoint blockade therapies.
Okay, that still feels backwards. If ICD is good, why is the gene signature linked to bad outcomes in immunotherapy resistance?
It's a puzzle, right? The signature included genes involved in immune regulation and inflammation, like FOXP3, often linked to regulatory T cells that suppress immunity, IL-6, an inflammatory cytokine, MYD88, involved in TLR signaling, like HMGB1 uses.
So maybe the signature isn't just ICD happening, but reflects a more complex, perhaps dysfunctional inflammatory state.
That seems to be the interpretation. The researchers suggested that these tumors might have high levels of immune modulation and escape mechanisms alongside the ICD signals. They saw high levels of immune checkpoint markers, for example. So the tumor might be undergoing cell death that should be immunogenic, but it's simultaneously putting up strong stop signs to counteract any resulting immune attack.
Like hitting the gas and the brakes at the same time?
Exactly. It really emphasizes that just inducing damp release might not be enough if the broader tumor environment is geared towards suppression.
So it's definitely not as simple as more ICD equals better. Context, again.
Context is king. Which brings us back to those complexities and contradictions we flagged at the start. HMGB1's role depends on its oxidation state where it is. ATP can be good, then bad if it turns into adenosine. HSP70 can be good in vitro, but linked to poor prognosis in vivo.
It really shows how much more we need to learn to reliably use these as biomarkers or therapeutic targets. You need to know which signal where, when, and in what context.
Absolutely. Study designs differ, patient populations differ, measurement techniques differ. All these things can lead to those seemingly contradictory results in the literature. We're still figuring out how to best measure and interpret these signals in patients.
Okay, let's try and pull this together for you, the learner. It's crystal clear that immunogenic cell death is a super active, really important area in cancer research right now.
Yeah.
Figuring out how therapies trigger it and finding reliable ways to measure it. That could honestly be game-changing.
Totally. Developing more effective, maybe more personalized cancer treatments hinges on understanding these processes and seeing how even within one cancer, like glioma, an ICD signature can predict outcomes and maybe therapy response. It just shows how tightly knit cell death and the immune system really are.
Yeah, and those confusing results around things like HSP70 and HMGB1, they're not just frustrating, they're reminders, right, that biology is complex, it's messy, and we've still got a lot of nuances to figure out.
Definitely. But the potential is huge. Seeing ICD-inducing strategies pop up across the board, radiation, chemo, targeted drugs, even viruses and heat, it offers so many potential avenues for boosting the body's own fight against cancer.
And looking beyond just classic ICD to things like necroptosis, ferroptosis, it just opens up even more possibilities for harnessing different types of cell death to stimulate immunity.
Which leads to the big questions for the future, doesn't it?
Yeah.
How do we get really precise about measuring and maybe even manipulating ICD in actual patients to get the best results?
And specifically for tough tumors like GBM, how do we overcome that built-in immune suppression so that ICD-based therapies can actually, work to their full potential?
Those are the multi-million-dollar questions, absolutely.
Well, thank you for joining us on this pretty deep exploration of immunogenic cell death. We really hope it's given you some valuable insights and maybe sparked your curiosity to dig even deeper into this really fascinating area.